Good afternoon, Sandberg. We're in chapter 4, so this is the first chapter after your first exam. You might be listening to this before your first exam, but please be studying for the first exam if you are. And if you have already taken the exam, then I hope you did well on your first exam and you're enjoying this class. So we're in chapter 4. We're going to be covering the membrane structure, so the different parts of the plasma membrane, and what it functions, so the movement of materials into and out of the cell. Plasma membrane is the outer border of all cells. Some cells do have cell walls, but not all cells do, and all cells have a plasma membrane, so I like to say that this is the outer border of all cells. And it's what determines the ability of other molecules to enter or exit. So it's a semi-permeable membrane that allows certain things in and keeps other things out. It's made up of mainly phospholipids. So there's a double membrane of phospholipids. The phospholipids, if you remember, they have tiny little heads that are made of glycerols. They are water-loving hydrophilic heads, so hydro-water-philic loving. And then they have water-fearing or hydrophobic tails that are sandwiched in between. So the heads, the glycerol heads, stick out into the extracellular and stick into the intracellular regions. And then the tails are sandwiched in between. Within the phospholipid bilayer, there are lots of proteins and there are cholesterols that help to stabilize the membranes. So here's what the plasma membrane looks like. These are your phospholipids. There's a protein. Some proteins are called integral proteins. Integral proteins span the entire membrane, so you can see them on the extracellular and the intracellular region. Some are called peripheral proteins. Peripheral proteins are found on only one side of the membrane and not the other. Peripheral proteins are easier to remove from the plasma membrane, but they still have really important functions. And then we can see some cholesterols here and here. Cholesterols help to increase the strength of the plasma membrane. Some of these proteins have carbohydrates on them. This carbohydrate chain makes the protein a glycoprotein, and we also have glycolipids. So proteins have a lot of functions in the plasma membrane. So the membrane itself is selectively permeable and only allows certain molecules across. So we have to have proteins that will carry other molecules or allow other molecules to move across freely. So a channel protein is a protein that molecules like ions, so atoms, ions, or can move from one side to the other side through diffusion. That would be facilitative diffusion. Carrier proteins bind with a molecule. Oftentimes these are like glucose molecules. They bind with the glucose and then they pull the glucose into the cell. Cell recognition proteins, these are your glycoproteins, and these can allow the organism to recognize the difference between a self cell and a foreign cell. And so typically they're like identification tags. So my cells have my own cell recognition proteins, which are like my fingerprints of my body. And my immune system won't attack my own cells. But if my cells got into someone else's body or vice versa, then the immune system would attack, which is why we have problems with like liver transplants, heart transplant patients, because we don't have the exact same cell proteins. You know, we can get close, but typically we're not identical. Receptor proteins are proteins that bind to some type of a receptor. So this could be a protein that binds a hormone. and then causes some reaction inside. And then we have enzymatic proteins, which act as enzymes, and enzymes increase the rate of a reaction by decreasing the amount of energy it takes for the reaction to occur. And so we have a lot of enzymatic proteins in our intestines because that's where the majority of breakdown of proteins and lipids and carbohydrates occurs. So the plasma membrane, in a nutshell, is a selectively permeable membrane. I think of it as a bouncer, and it basically allows certain molecules in and keeps other molecules from getting in. So the ability to move directly across the membrane will be dependent on the size of the molecule and if the molecule is charged or polar or not. If the molecule is charged, so it's a polar molecule, or if it is very large, it's not going to move across the membrane. Then it's going to have to find another way to get in, either through facilitative diffusion, where it moves across a protein channel or carrier protein, or through an active mechanism where it is using energy to pull the molecules in. So we're going to talk about the different mechanisms to move molecules into or out of a cell. But first you have to understand what a concentration gradient is. A concentration gradient is the amount of substance that is found on one side of the membrane and the amount found on another side of the membrane. The concentration, so if you're talking about molecules or ions or anything, you want to have equal concentration on both sides. So molecules will follow a concentration gradient from higher to lower concentration naturally until they're at equilibrium. You don't tend to go against the concentration gradient unless you are using energy. You're using energy to push things against that gradient. I always think of clown cars. So if you have a high concentration of clowns inside of a tiny little car like a Datsun, as soon as the doors open, clowns start to pile out. And they move out, out, out until you have completely emptied the car and the clowns are all over the place. And that would require no energy. Clowns are just falling out because there's so many inside. If we want to pack the clowns in, it's going to take energy to push all the clowns in and get them put in the proper way. So that requires an input of energy. So here you can see a figure from the textbook, shows charged molecules like ions don't tend to go across the membrane. Large molecules like proteins don't go across the membrane. Non-charged and small molecules can go directly across. And water, which is a special type of molecule, it is considered a polar molecule, but water is our only special molecule that still can go directly across the membrane. Even though it is polar, it can go directly across. So it breaks all the rules. Though it does go across the plasma membrane, most water still goes through protein channels called aquaporins. So we're going to look at energy or non-energy required movements, so passive transport mechanisms, and then we'll talk about active mechanisms. So passive transport, we need no energy. And the first is just basic diffusion, simple diffusion, where molecules move from higher to lower concentration down a concentration gradient until they reach equilibrium. So here we have all of these lipid-soluble molecules, whatever they are. And they're going directly down the concentration gradient from higher to lower concentration until they hit equilibrium. I like this picture because it shows what's going on. Say you took some dye and you put it into a beaker filled with water. The dye is going to sit at one spot and it's going to slowly start to diffuse outwards. And it will continuously diffuse outwards until we have an equal distribution of water and dye ratio. Think of your Kool-Aid. Have you? I am sure that you've all either made Kool-Aid or you've seen Kool-Aid getting made. And when mom puts the Kool-Aid in or dad puts the Kool-Aid into the water, initially the Kool-Aid is found in like this bright red at the very bottom of the container. And then you mix it around and it slowly starts to cause all the other regions of the water to start turning pink or red or whatever color you're making of Kool-Aid. That's simple diffusion.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Osmosis is the diffusion of water across a selectively permeable membrane. And again, we're moving water from higher to lower concentrations, and typically water moves through their aquaporins, but they can move directly through the lipid bilayer as well. Osmosis moves from higher to lower concentrations of water until you have equal water to solute ratios on both sides. And so here we're looking at a beaker that contains 70% water and 30% solute. We have a container inside which has a selectively permeable membrane, and the fluid inside the container contains 60% solute and 40% water. So each of them contains 100% of something, right? So water is going to move in both directions, but water is going to move faster either into or out of the beaker, depending on the amount of solute and water inside the container. And since here we have 70% water and 30% solute in here, and then 60 and 40 here, there's more water here, less water here. Water is going to move much more rapidly into the container, into this container, until we have an equilibrium of equal water solute ratio in the beaker and water solute ratio in the this container. So over here you have the same thing, semi-permeable membrane, but now we have 70% water and 30% solute, and 70% water and 30% solute. So the amount of movement of water is going to be in both directions, so you'll always have movement of water in both directions, but the amount of water moving into this container and out of back into the beaker will be equal, because there's not going to be movement in one direction or the other. They're at equilibrium already. There are three types of tonicities that we talk about. Iso means the same as, and so an isotonic solution has an equal amount of solute to water ratio inside and outside of the cell. In a hypotonic solution, there is less solute in the solution than there is in the cell. And so in a hypotonic solution, we have actually more water outside of the cell, and so water moves into where we have more solute in the cell. In a hypertonic solution, there is more solute inside the cell, or I'm sorry, I said that wrong, more solute outside the cell, so in the solution outside the cell, than in the cell. And so the cell actually contains more water, and the solution that the cell is in contains less water, and so water will leave the cell. I like to think of these in an isotonic solution. You'll have equal amounts of water moving in and out, so you're not going to have a gain of water on either side. But in a hypotonic solution, you have more water outside of the cell and more solute inside of the cell, and so water enters the cell, and this causes the cell to swell. So I always think of hypo as hippo, and hippos are very large animals. And so if you have a hypotonic solution, your cell's going to get very large. In a hypertonic solution, there's more water in the cell and less outside, and so water is going to leave the cell faster than it enters the cell. And this causes the cell to start to shrink or shrivel up. That's called crenation. So when I think of the word hyper, I think of little kids, because little kids are very hyper, and they're little. So in a hypertonic solution, the cell gets very little. It loses water and it shrinks. And these are blood cells. This is a happy blood cell. It's in an isotonic solution. Here's a hypotonic solution on blood, and this causes blood cells to burst. The term of cell bursting is cytolysis. The term for blood cells, red blood cells to burst, is hemolysis. And then here's hypertonic solution. This is blood in a hypertonic solution. The blood will actually shrivel up. Plants have a different response to the different solutions. In an isotonic solution, the plant's not going to gain a lot of water, and it's not going to lose a lot of water. The plant tends to not be super happy. It'll kind of droop a little bit. It's pretty happy when it gets into a hypotonic solution where the cells gain water. The plant has this thing called turgor pressure on the walls, and it keeps the plant upright, and the cell wall actually prevents bursting. And then we have hypertonic solution. Cell loses water, so in this case, the cell, because we have cell walls, the walls don't change in shape or in structure, but the plasma membrane shrinks away from the walls of the cells. And that's called plasmolysis. Facilitative diffusion is movement of molecules down a concentration gradient through either a protein carrier or a channel protein, allowing molecules to move from one side to the other side of a membrane. So here's an example of, it's also a passive mechanism. We're moving from higher to lower concentration. We're just using a protein carrier. And so in this case, we have this carrier protein binds this molecule, and the binding allows it to open to the extracellular region or the intracellular region, and the molecule gets inside. If this was channel proteins allow ions to just move across them from higher to lower concentration. Active transport then uses energy that allows molecules to move either down their concentration gradient, but the molecules are too large, so they can't go directly through the plasma membrane, and so we need to use like a mechanism to pull them in or push them in, or they're moving against a concentration gradient. So we have to use energy to get them to move across the membrane. But we're using energy, and typically the energy that we use is called ATP. Molecules, or in a pump, a solute pump, molecules combine to or bond to a carrier protein, and then ATP binds, and then the molecules are going to be actively pushed against their gradient, either outside or inside of the cell, wherever they're going to go. And the ATP drives this motion. ATP is hydrolyzed to the ADP plus the phosphate, and that pushes the molecules across the membrane. So here is a sodium-potassium pump. This is one of the most famous pumps we actually have. We have them. A lot of bacteria have these pumps. And so what happens is sodium binds to the pump in its resting form. And so the sodium is found inside the cell. It moves in to the cell through these channel proteins that are gated. And then we need to push the sodium back out. So sodium there's more sodium outside of the cell than inside the cell. So we want to push the sodium that's in the cell out of the cell against its gradient. Sodium binds to this pump, and ATP binds. It's hydrolyzed to an ADP plus a phosphate that activates the pump, and the pump opens to the extracellular environment. Sodium leaves. Now potassium to potassium combined. When the phosphate releases from the ATP binding site, when the phosphate releases for the ATP binding site, that allows the protein to go back to its resting membrane or resting structure, and it opens to the inside, and then potassium is released to the inside of the cell.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
We also have vesicular transport. Vesicular transport is an active mechanism in which the cell itself either pulls molecules in through a process called endocytosis, or releases molecules through a process called exocytosis. And so the plasma membrane is going to form a vesicle around the molecules, or molecules inside the cell. A vesicle inside the cell binds to the plasma membrane to release molecules. So the first mechanism is called exocytosis, and exocytosis is when we produce a vesicle inside the cell, and that vesicle is going to release contents into the extracellular environment. The vesicle binds to the plasma membrane and actually becomes part of the plasma membrane, so the membrane itself gets larger. It opens up and materials are exiting the cell. So exocytosis, we're exiting the cell. Here we have this plasma membrane bound vesicle. The vesicle binds to the plasma membrane and actually becomes part of the plasma membrane, and it releases materials into the extracellular environment. Endocytosis then is the movement of materials from the extracellular environment into the cell. There are 3 types of endocytosis. I always talk about phagocytosis as cellular eating. So you're taking in large molecules, large materials. This is like where your white blood cells pull in bacteria and break the bacteria down. Penocytosis is cellular drinking. Almost all of our cells can go through penocytosis, and this is where all the cells take in tiny droplets that have dissolved solutes in the solution. And then we have receptor-mediated endocytosis. This is a special form of endocytosis in which we take in specific molecules. And the molecules that we typically talk about are cholesterol molecules. And so here we have phagocytosis, where you have a large substance being taken in. We produce a vesicle that surrounds the whatever this is, the molecule, the food or whatever, and then the vesicle blebs off into the cell, and so the plasma membrane gets smaller. Here's penocytosis, where we're taking in small droplets, and then receptor-mediated endocytosis, where we have these receptors that are embedded into their protein receptors that are embedded into the cell itself in the plasma membrane, and molecules, in this case these would be cholesterol molecules, bind, and once all of the receptors are bound, the entire region folds in on itself, and via penocytosis, we bring those molecules into the cell. This is how we get rid of most of our cholesterol. And so we all know cholesterol is bad. If these receptors don't bind properly, then we don't get rid of all the cholesterol, and that can cause problems like high cholesterol and diseases associated with it. All right, I'm going to stop here because it's the end of the chapter, and I will see you on Monday night. Bye.